In this paper, we present DeepSee, an interactive workspace for studying deep ocean ecosystems. DeepSee visualizes sediment core samples collected during deep ocean field expeditions in multiple dimensions across 3 coordinated views. The map view plots previously sampled cores as a scatter plot using latitude and longitude on top of maps representing geological landscapes and surface biological and ephemeral soft features. Users start by filtering cores by named location, date range, and core fate. Users then navigate by clicking and dragging to pan and zoom. Cores remain the same size at all levels of zoom to help users track them as the view changes. We provide several details on demand. We label the plot with latitude and longitude grid lines and convert the width and height of the current viewport into metric units. This gives users a baseline for the size of the geological and biological features they see on the map, as well as the distance between sampled cores at all times. On hover, users can see the named location, fate, date, and exact latitude and longitude of any core. Users can also switch the map on the fly, and the boundaries of the viewport will persist, helping users maintain context as the view changes. Once a site of interest has been located, we support evidence marshalling through annotation with an in situ drawing tool on the map with 6 colors and undo and redo capabilities. The user naturally concludes their exploration by selecting cores to visualize in the core view or interpolation view. The core view arranges columns of horizontal bar charts that visualize the sample data selected in the map view. The bar charts show core IDs, fates, dates, and are aligned vertically by depth horizon to facilitate comparison across bar charts along the same horizontal baseline. Because our data consists of hundreds of geochemical and biological parameters, users can search for a specific attribute from a drop-down menu. They also have a choice of 6 different colorblind-friendly color palettes. Finally, users can deselect cores from this view to narrow the space of cores for subsequent analysis in the interpolation view. The interpolation view visualizes sample data by interpolating geochemical and biological parameters between cores in 3 dimensions by latitude, longitude, and depth. We compute and render the 3D interpolation data in real time as space-filling volumetric pixels, or voxels, colored by the parameter value of each sample at every 3D point in space. Using a mouse, users can left-click and drag to freely rotate the voxels with a stationary camera angle, right-click and drag to translate the camera in the latitude-longitude plane, and scroll the mouse wheel to zoom in and out. DeepSee currently supports fast 3D linear barycentric interpolation and a 3D discretized approximation of Simpson's natural neighbors interpolation. The grid sizes offered were specifically chosen to mirror the real-life sizes of cores, which are 7 centimeters in diameter. Because interpolations are computationally expensive, we provide larger grid sizes in 7 centimeter increments. In this view, we also offer the same 6 accessible color palettes. We also implemented variations on the 6 standard color palettes based on value-suppressing uncertainty palettes, or VSUPs. To do this, we normalize the linear distances from each voxel to the nearest non-interpolated voxel and use that as uncertainty. Thus, interpolated values farther from a known sample value will appear suppressed, signaling a lack of confidence in the interpolation at that location. The color palette can be changed on the fly while preserving the camera angle and visual embellishments. As users explore the interpolation, we provide several on-the-fly capabilities to help users maintain spatial context. We plot the rectangular convex hull of the selected points on both the map view and the interpolation view with a white arrow always pointing north, helping users spatially orient themselves. Users can reconfigure the view in several ways using buttons. One, by extending the height of each voxel from 1 centimeter to 10 centimeters, making patterns in depth much easier to see. Two, showing and hiding all interpolated values and only showing actual sample data. And three, changing voxels from rectangular prisms to cylinders fixed at a 7 centimeter distance, mirroring the shape of real-life cores. Users can also clip through the voxels to see patterns in the data in the interior of the interpolated region. We provide two clipping methods. One, clipping individual voxels by their interpolation value outside of a range chosen by the user using a double-ended range slider. And two, clipping by latitude, longitude, and depth using a single-ended range slider. Finally, users can select a vertical core to save. The results are output in JSON as a list of parameter values at each 1 centimeter depth, matching the exact format of a real core sample. These results can then be exported and further analyzed in other software. For a detailed breakdown of the design process, usage scenarios, and an evaluation with ocean scientists that deploy DeepSee on a real expedition, please read our paper. Thank you.